water now, okay? Thinking ahead to photosynthesis, what's going to happen to the water in the light reaction? Light reaction, what's going to happen to the water? We're going to split it. We're going to make oxygen, but what's going to happen to the electrons? The electrons are going to get more energy in the light reaction, and those are eventually going to end up in the sugar. And then in respiration, we're going to drop those electrons down to make ATP. And then in photosynthesis, we're going to store the energy back. Does that make sense? And water's huge. It's right in the middle of this cycling, back and forth and back and forth. Questions on this slide, because I'm about to switch it. Any questions on any terms? Yeah?
This is kind of back to level 12.
Right.
What's the difference between entropy and enthalpy?
Yeah. Entropy is randomness. Enthalpy is total energy. And so glucose has more energy than carbon dioxide and water. And so the total energy is going to drop. But the entropy is going to go up. And the way I avoid that confusion of those two terms, because they sound the same, is I just think of enthalpy as total energy in the system. Just call it that. Yeah, tough.
So ATP synthase is using the power of diffusion, right?
Right on. Yeah. And so are we good that this is facilitated diffusion? It's moving through. But what's ATP synthase using that energy to do? To phosphorylate. In other words, to add a phosphate again. From, how did everything move? That's a really good question. How does everything get where it needs to go? Diffusion, diffusion, diffusion. Okay? Everything's moving from diffusion, right?
It got carried there by now.
Yeah, how did the phosphate get here? It moved there through diffusion. Why is it going there? Because all of it's being gobbled up by a black hole of phosphate-eating ATP synthase. Right there, the phosphate disappears. It's not there anymore. What's there in its place? ATP. So where's the ATP going to go? It's going to go from here, where there's a bunch of ATP, to anywhere else in the cell where it's needed. Because the ATP there is going to be gobbled up. So everything is moving by diffusion. Are you with me? All right, let's go to the next one then. So this is photosynthesis. Two parts. Are we good on thylakoid, stroma? Thylakoid light reaction. Stroma is going to be the Calvin cycle. What's going on in the light reaction? Splitting water to make oxygen. Where's the energy go? Into NADPH. Where does it go after that? It's going to eventually end up in the sugars. And so we're going to get that electron and the hydrogen very excited, and we're eventually going to transfer that to sugar. Let's go dig a little bit deeper into the light reaction. So there are two photosystems, 2 and 1. Which photosystem is responsible for making ATP? I would say photosystem 2. Can you see it? Electrons down the electron transport chain, a bunch of protons, they go out and they make ATP. Which photosystem is responsible for making NADPH? Photosystem 1. Why do we have this before that? Because we really have to bump up our electrons. So the electrons have low energy in water, and so in photosystem 2, we get them excited and eventually transfer a lot of that energy to ATP. They already have more energy here, and now we can boost it even to a higher level to make NADPH. Do you remember the scaffolding with the guys with the hats and their hammers and everything? Okay, yeah, so it's a double boost. Questions on this before we go? All right, you guys are doing good. Let's go to the Calvin cycle. What are we doing? We're taking in carbon dioxide and adding energy to it. The energy comes from the ATP, but it also, the high energy protons come from the NADPH. And so what do we make at the end of Calvin cycle? What's down here? G3P. Not glucose, but G3P. Do you remember that we can then make G3P into the glucose inside the plant? Questions on photosynthesis? Are we good? Keep going. Okay, so let's keep going. So that's it for 12, 13. Now let's go to polymers. Anybody watch the videos on polymers or some of the stuff that I've added? So if you're confused on anything I'm about to run through, there are videos on each of these. Let's start with polymers. Polymers are made up of monomers. Okay, so let's play the polymer-monomer game. What are the 4 types of macromolecules? Number 1, carbohydrates, lipids, proteins, nucleic acids. Are we good? We can do all those? Let's go carbohydrate. That's a polymer. What's the monomer? Sugar, or monosaccharides. Yeah. Let's go polymer protein. What's going to be the monomer? Amino acid. Let's go the protein, or the polymer nucleic acid. What's the monomer? Nucleotides. And then lipids don't have those monomers. And so polymers are made up of monomers that are attached together. So how are they attached chemically? So this is a dehydration reaction. What am I attaching together here? Let me pause for a second. look at it for a second. What am I attaching together? Two, what do you say, Marin? Right, but what are these guys? What are these monomers? Right, amino acids, right? So we're going to attach them together, and we do that by taking off a water and then attaching them together. Well, that's a dehydration reaction. What's the opposite of that? Can you see this is a disaccharide? So we have 2 sugar. What am I going to add to it? A water, and that's going to split it into 2. Now let's step back for a second and go big picture. Where in my body am I going to take giant polymers and break them down into monomers? Where would I do that? Anybody, a hand? Yeah, where? Right, so as I go through my digestive system, I'm taking my food and I'm chopping it up. Okay, let's say I have starch. I have spaghetti last night. That starch, I'm going to chop it up into all its little monosaccharides. Where am I going to do dehydration? Where am I going to attach these monomers together again? Where would I do that? Where do I build? Yeah, what do you think it's up? In the cell, when I want to make something. Yeah, okay. And more specifically, let's say we're making a protein, it'd be in the ribosome itself. Does that make sense? Are we all good that you are what you eat? Like what you're looking at right now is food that I ate, and most of it's really gross because I like high fat, high sugar. I try not to, but I do. And so when I chop down proteins, I eventually build that back to make meat. Okay. So polymers, monomers. Questions there? Are we ready to fly through the different macromolecules? Let's start with sugar. These are the carbohydrates. What's the building block? A monosaccharide. This is the empirical formula. It's the ratio of atoms, so 1 to 2 to 1. Keep an eye on this as we go through it. That number is going to get bigger. All these numbers are going to get really big. So let's start with the first one, monosaccharides. Our example is going to be glucose. You know, we use it in respiration, we produce it in photosynthesis. Here's a couple of other monosaccharides, fructose and galactose. A lot of us love high fructose corn syrup, right? Yeah, so that's going to be a monosaccharide. What's cool about these 3 is that they can flow through our blood and they can move directly into the cells, not the butt. Yeah.
Are we supposed to know all the...
Everything, you got to know everything. But yeah, you're right. I'm not going to ask you to draw all the structures of that. Yeah, absolutely.
Just like basically glucose, should we know?
We should know a monosaccharide, disaccharide. Yeah. Yeah. The only one that we really should know, you know, glucose, C6H12O6, we should know that structural formula, or the molecular formula. But all these have the same molecular formula. All of them are C6H12O6. They're just put together in a different way. Yeah, tough.
It's like the hardest level.
Of the stuff we did, I think so. Yeah. Don't you think so? Yeah.
Or I'm not even like
hardest unit?
whole.
I would say the test scores on this test are probably lowest of everything we do all year. Yeah. And it's not that the content, it's just it's so much content. But I also prepared you more this year than I think I ever had. I think we did a better job. So we'll see how it goes. Yeah. I think we'll do better than last year. Yeah, Eric.
What was like last year's average score?
I don't know. It was like, yeah, 8 or 10% lower than the other ones. Yeah. I mean, you could talk to other clients. They'll tell you that. This is a hard one. Yeah. So disaccharides are next. Can you see what happened to my number? Doubled it. So we're going to have 2 monosaccharides attached together. That's table sugar. That's lactose, disaccharides. And then if we were to look at polysaccharides, that's going to be, I mean, look at the numbers here. So this is starch. It's 2 monosaccharides on either side, and then there's going to be hundreds of sugar molecules on the inside. So over. And so we have to chop that up before we can get to the monosaccharides. Colleen.
How are the 2 on this side different from all the ones in the
Yeah, we're going to lack, so what is the big difference? We're going to have you're dangling a hydroxyl group off the end. Can you see that on the sides? Yeah. And this is a really good question. So let me ask you a quick question. Let's say I have a disaccharide and I want to break it in half. How many water molecules am I going to require to do that? So if I have a disaccharide, how much water is required to break it in half? 1, right in the middle, right? So let's split it in half. Let's say I have a protein that's 50 amino acids long. How many water molecules am I going to require to break that down into all its individual amino acids? 49, right? Because there's going to be 50 of them, and between each of them, we're going to have to add a hydroxyl group, or a water. Yeah. And so that's the big thing, because we have that bond. Yeah. Let's go to the next one. This is glycogen. What's that? It's polysaccharide in U. So you take sugar in, you chop it into monosaccharides, and then you store that as glycogen. And so we store a lot of the glycogen in our liver. If I need sugar, then I can secrete enzymes, and so insulin and glucagon are going to work to shuttle that sugar between the liver and the glycogen in your cells. We'll get that in the next level. And then also don't forget on sugar that we can get structure out of it. So the cellulose on your paper or the cellulose in a tree is going to be polysaccharide, but so is chitin. Do you remember that word that we all called chitin? But it's chitin, and it's found in the exoskeleton of insects and in fungi. Let's go to lipids then. The major lipid is going to be a triglyceride. It's got a glycerol head and then fatty acid tails. But there are two other important ones. Phospholipid cholesterol, are you good now with what they do, since we've gone through 15 and 16?
We need to know the, I mean, like, is it?
You got to know everything, yeah. But let's say what are the big parts? There's a glycerol head, and then we're going to have fatty acid tails.
So it's just 3 instead of 2 for a phospholipid?
Right, but what's the other big difference here?
Yeah.
There's a phosphate group, and that's going to cause the head to be polar. Yeah. Let's go through specifically, where's the energy in? Again, between the carbon and the hydrogen, and those electrons between the two. And so that's why those fatty acid tails are hydrocarbons. But so is gasoline, and so is going to be candle wax.